Welcome to our video lecture on transportation in the phloem of plants. Our essential understanding for today is that structure and function are correlated in the phloem of plants. Our objectives, we're going to define translocation, which is the function of phloem. We're going to outline phloem's structure, how it helps do translocation. We're going to describe sucrose loading. We'll review osmolarity. We're going to describe how aphids, which are bugs, can be used to study the rate of translocation, and this is like a super weird thing that scientists do. It's kind of funny. And then we're going to compare and contrast xylem and phloem. So translocation is what happens in phloem. It is the movement of organic compounds from source to sink. We're going to talk mostly about sugars, and when I have sugars dissolved in the plant, we're going to call that often sap. So sap or sugars moving through the phloem from source to sink. Those sugars are mostly going to be transported as sucrose. Sucrose is a good option because it's quite soluble. It dissolves in water so that we can actually make that sap, that syrup. But it's also quite stable. It doesn't get sucked into mitochondria and turned into carbon dioxide used to make ATP too fast. So this idea of source and sink. Remember that our xylem is what transports water and minerals. It's always going to move up, always up the plant. So xylem can only move in one direction. We're going to move up that plant from the root through the stem up to the leaves. So xylem in transpiration is water and minerals always up. Our phloem, moving these saps, these sugars, moves from source to sink. And what's interesting is that the sources can kind of flip around a little bit. The sinks can flip around. So if we are talking about the source, this is where the sugar is starting, and it might be where we're making those sugars in photosynthesis. So it could be some mature leaves, it could be some mature stems where we're making the sugar. Then maybe we send the sugar down to the roots, and the roots use the sugar to make ATP so that they can do all that active transport of moving those ions around so that we can get water into the root. We also might send some of those sugars down into a taproot or a tuber where we're going to store that sugar. Or we might need to send that sugar to grow some leaves. But these roots and tubers, they're going to store the sugar. At some point, I'm going to need to use that sugar. And when we do that, those taproots and tubers, those seeds actually become the sources of the sugar. So if I'm taking the sugar out of the potato, out of the tuber, and I'm using it to grow new leaves, well, now the source is the tuber and the sink is the leaf because I need the sugar here to grow the leaf. And then once the leaf is all grown up, the leaf will go back to being a source and we can send that sugar back to the potato. So sources and sinks can kind of flip around a little bit. So we can move that sucrose up. We can move that sucrose down, which is different from the water in the xylem. That water always moves only up and out. So the phloem's structure is ideally suited to carry out its purpose, its function of translocation. Two kinds of cells in phloem. We have sieve tube cells. Sometimes we call them sieve tube elements. And there are also companion cells. Those sieve tube elements are separated by sieve plates. So the cell wall between them is modified. It has a lot of holes, which just makes it easy for that sucrose, that sap, to move back and forth between the cells. The sieve tube elements are living, which is different. We can remember that xylem cells are all dead. Phloem cells, these sieve tube element cells, are living, but just barely. They need a lot of support, and they get that support from the companion cells. They don't have a nucleus. They have very little cytoplasm. They are shells of cells, if you will. Those companion cells that sit right next to, so here's a companion cell, sieve tube element, companion cell. Those companion cells that sit right next to the sieve tube elements, they are connected, and their job is to not just support those sieve tube elements, but also to actively transport sucrose into the sieve cells. Here's just another look at our sieve cells and our companion cells. So here's the sieve plate, that modified cell wall between different sieve tube elements, lots of pores, lots of holes so that the sucrose, the sap, can move super easy back and forth. We have next to those sieve tube elements, we have those companion cells. Their job is to load lots of sugar into those sieve tube cells so that that sugar can move around the plant. So I've mentioned this idea of sucrose loading a couple times now. Sucrose loading is where we actively transport that sucrose, that sugar or sap, into the sieve element cells using the super cool thing called a co-transporter. It is active transport. We need some ATP, but we use it in kind of a weird way. What we're going to do is use a co-transporter protein. That's this guy here. In order to activate that co-transporter protein, we're going to need to pump some hydrogen ions. Now you're having photosynthesis flashbacks. We're going to use the ATP to pump the hydrogen ions out of the phloem cells into the apoplast. That's that space between the cell membranes, the cell walls of the plant cells. We're going to pump those hydrogen ions out into the apoplast. Now I have this crazy high concentration gradient of hydrogen ions, which the universe doesn't like. And so these hydrogen ions are going to flow through, not ATP synthase, but this time we're going to use that co-transporter protein. So the hydrogen ions are going to flow through that co-transporter protein from high concentration to low concentration to get back to equilibrium. What's interesting about this co-transporter protein is that it can only move hydrogen ions with a sucrose molecule. So we're going to take this sucrose molecule, and this time we're actually moving the sucrose from low to high concentration as the hydrogen ions go from high to low. And so I'm going to take that hydrogen ion, it's going to move back into the phloem cells. The sucrose is going to come with the hydrogen ions, and we're going to have a higher concentration of the sucrose inside the phloem cells. This is sucrose loading, active transport. We do need that ATP. We're moving the sucrose from low to high concentration gradient, so against its concentration gradient, but we're doing that while we move the hydrogen ions along their concentration gradient from high to low. Crazy cool, right? This sucrose loading leads to lots of pressure inside the phloem. We call it hydrostatic pressure because we've got, of course, water involved. So what happens is we have this source, this leaf cell. It's going to make some sugar. It's going to get loaded into the companion cell. The companion cell is going to do that hydrogen ion sucrose loading thing with the co-transporter protein. We're going to end up with lots and lots of sugar molecules, all these sucrose molecules in the sieve tube elements. And what happens is now I have a hypertonic environment here. So much sugar, which means that water via osmosis is going to move from the xylem, which is right next door, into the phloem. And so now I have not just sugar, but also lots of water, and sugar and water, and it's filling up all this space and more sugar and more water, and the pressure is very, very high. It needs to move away from this high pressure. And it could move down or it could move up. It's going to depend on where we have lower hydrostatic pressure. Where we have that low pressure is actually where I'm pulling the sugar back out of the phloem. And maybe that's happening here in a root cell. Maybe here I have the root that needs to use the sugar to pump the hydrogen ions to get the phosphates in so that we get that mass flow of water into the root cells. So the companion cells are actually going to pull the sugar out of the sieve tube elements and pump them, those sugar molecules, into the root cell so the root cell can make lots of ATP to do all of its work. Because I'm pulling sugar out here, this environment becomes hypotonic, which means that the water molecules can move back over to the xylem and maybe do this all over again, or perhaps leave through the leaf. But because this is lower pressure, because I moved out the sugar and the water, there's less stuff here, there's more space. All this stuff that's up here all squished together will move down to where there's more space. And then the companion cells can move it out, the water will move back into the xylem, and it can happen all over again. Now, if I had, instead of the root being the sink, let's say that we were growing a new apple up here. Ooh, that's an apple. Let's pretend. I think I made it worse. Okay, there's an apple. I'll label it so that we know that's an apple. Perhaps we need the phloem to send the sugar up to the apple so that we can grow this new baby apple, load it with lots of sugar. And so the companion cells would be pumping the sucrose in here. The water would again go back to the xylem, or maybe stay in the apple with the sucrose. And then this would be our low pressure because we're pulling the sucrose out of the phloem up here to grow this apple. Because the low pressure is here and the high pressure is still here, that sugar would move up toward the apple to where we have lower pressure because there's more space. And so of course it would move to where there's more space. So this hydrostatic pressure just allows the sucrose in the phloem to move from where there's a high pressure to a low pressure. And that low pressure might be in the root, but that low pressure might be up in an apple. So the sucrose can move in multiple directions, just depending on where I have that high concentration of sugar, which pulls in some water from the xylem because of osmosis, where we have those lower concentrations of sugar. So just a quick reminder about this idea of osmosis and tonicity, osmolarity. We know that our animal cells like isotonic environments. They like equal inside and outside the cells. That allows for dynamic equilibrium. Water is still moving in and out of the cells, but it's moving at equal rates, and so the cells are nicely sized. In a hypotonic environment, those animal cells will get too much water and they could lyse. They could explode because they have too much water. But that hypotonic environment is super happy for plants. We call them turgid. They're nice and full and fluffy of the water. Hypotonic, remember, hypo rhymes with low. We have lower concentrations outside, higher concentrations of stuff inside, so the water moves into the cell. No one likes a hypertonic environment. Hypertonic is where we have more stuff outside, less stuff inside the cell, and so water will move out of the cell into that salt or that sugar, that protein, whatever is that's outside the cell. This is really bad for plant cells. The cell membrane actually starts to pull away from the cell wall. They're super unhappy, and we call that plasmolyzed. The animal cells will lose volume because of that loss of water. We call that crenellation. So, I mentioned at the start, one of our objectives would be to talk about how we use aphids, these bugs aphids, to measure the rate of translocation in phloem. This is a crazy thing that we do. So aphids are basically the mosquitoes of plants, and they have these crazy long projections called stylets. And these stylets get sent down into the phloem cells. They suck the sap out of the phloem, just like mosquitoes suck the blood out of our blood vessels. And this is how the stylets eat. So here's an actual aphid, and he's got his stylet down into the plant. And aphids and their stylets are built so that the stylets end right at the phloem. We scientists, with needles or pipettes, we don't know when to stop at the phloem, not nearly as well as the aphids do. So scientists decided that it was way easier to let an aphid stick its stylet into the phloem. Easier to do that than for us scientists to try to stick a needle into the phloem. The aphid will stick its stylet into the phloem, and then the scientist will use tiny little scissors and cut off the stylet. We just chop it right off. And then here's the stylet in the plant. Here's the stylet in the plant. Poor aphid runs away because it lost its stylet. So here's the stylet. Because in the phloem we have that crazy high pressure of sucrose, that sap will ooze up and out of the stylet that just got cut off. And then scientists can analyze how fast, how much, what is the composition of that sucrose in that stylet. And then we can collect some super cool data. So let's say that we have our plant here. We added some radioactive carbon dioxide. We know that in the Calvin cycle that radioactive carbon dioxide will get incorporated into the sugars. So then the plant is using that CO2, that radioactive CO2, to make radioactive sugar, and that sugar starts to move through the phloem from the source to the sink. And then we have one aphid here, we have more aphids lower, we have more aphids lower, we have more aphids lower. And we don't just measure that distance from the chamber to the colonies, but we also measure how long it takes for the radioactive carbon to get to the different colonies. And so maybe we start making that CO2. It takes an hour, 1.25 hours, hour 15 minutes for that radioactive carbon to get from here to here. And then it takes this long to get from here to here. We can use that to measure the rate of the phloem because we know the distance between the colonies. And it's super cool. And we do this with bugs, and then we cut off their noses. They're not noses, they're stylids. But crazy cool, right? All right, so last little piece of the day, we're going to review today's lecture and the previous lecture on xylem with a little comparison and contrasting of xylem and phloem. So what is the name of the transportation method of xylem? In xylem, it is called transpiration. And then in phloem, it's called translocation. Not to be confused with transcription or translation, transpiration, translocation. Good. Transport direction. In the xylem, it's always going to be up from the root through the stem to the leaves. In the phloem, it can be multiple directions, but we'll always go from the source to the sink. But the source can be different and the sink can be different. We might be going up or down. What are we transporting in the xylem? It's always going to be water and minerals. And then in the phloem, it's going to be organic molecules. We talked mostly about those sugars, sucrose. We call it sap. It's how we make maple syrup. Active transport or passive transport. In the xylem, we do have a little bit of active transport in the roots to move around those ions, and then we have osmosis happening, and then transpiration itself is quite passive due to that water potential. In phloem, we have active transport in the companion cells to load that sucrose, but then once the sucrose is in the sieve tube element cells, the rest of it is passive and depends again on pressure, on that hydrostatic pressure of the sap in the phloem. Structure, xylem is completely dead. We have just cellulose cell walls and that lignin that helps to support the cellulose. In our phloem, we have two kinds of cells. We have the sieve tube element cells, and then we have the companion cells. These guys are almost dead. But not quite yet. Companion cells are quite alive. The sieve tube element cells have special cell walls called sieve plates between them that allows that sucrose to move super, super easy. In our xylem lecture, I talked about how to find xylem in the micrograph pictures that you might see. Xylem cells are always going to be bigger. So all of these guys, xylem cells, each one of these clumps we would call a vascular bundle. Vascular bundles have the xylem and then also the phloem. So phloem would be up here. In between the xylem and the phloem, we have some cambium. These are the stem cells, the meristem of the plant. So we have phloem. Phloem is always going to be closer to the outside of the plant, so closer to the stem, the outside of the stem. Xylem will be closer to the inside of the stem. So xylem here. And that's hard to read that. Xylem here, phloem up here. Good. We did it. We accomplished our objectives. We defined translocation. It's that movement of sucrose from source to sink. We outlined phloem structure, two kinds of cells, both living, but one just barely living, those sieve tube elements, and then the companion cells. Sucrose loading happens with a co-transporter protein. We're going to pump the hydrogen ions out of this cell. The co-transporter protein will bring the hydrogen ions back into the cells, but it's going to bring sucrose with it. We reviewed osmolarity, the hypertonic, isotonic, hypotonic. We described those aphids and how we can cut off their stylets, and then the sap will ooze out of them. We can use that to study the rate of translocation. We wrapped up with our little comparison and contrasting of xylem and phloem. Well done today, my friends.